Good afternoon. Welcome back to UCSF Department of Medicine Grand Rounds. I'm Bob Wachter, chair of the department. Welcome, as usual, to our live audience from our department, to our other colleagues at UCSF and throughout the UCSF Health Network. As usual, we will post this to YouTube tonight at about 7 o'clock or so. You see the address there, and I will tweet out a few summary comments as well as that web address at that time. And again, we're gratified that this series has been so useful and is now up to about 1.1 million views over the course of our series. A few quick ground rules are seen here. You're used to these by now. Put your Zoom window in full screen mode. Everyone's on mute. If you have questions, please type them into the Q&A box and we will get to as many as we can. And you see the address for the YouTube video later. Well, we find ourselves in an amazing moment of this pandemic. And as I said on Twitter last night, it feels like we're viewing this through a split screen. On one, we're witnessing a horror movie of really epic proportions now, with 250,000 dead people in the United States, cases skyrocketing around the nation, hospitals filling up, and some of them beginning to become overwhelmed. We've continued to have a relatively benign course in San Francisco, and knock on wood, that will continue, but our case rates have gone up about threefold in the last few weeks, and hospitalizations and deaths are sure to follow. Hopefully we can turn this around as we have before, but we will see. Really no place in the United States, or really for that matter for the world, seems safe. So that's what we're seeing on one half of the split screen, which is incredibly distressing. And on another half of the split screen feels like a feel-good rom-com where we're getting news sort of seemingly daily that's really, really good news. In fact, the news on vaccines is actually quite remarkable, almost beyond the dreams that we might have had a few months ago. And we've had some good news on therapeutics and testing as well. So today we're going to turn to the two areas of good news, vaccines and therapeutics, in the context of the current tragic situation that we're seeing. What does the vaccine data mean? What's next in terms of the FDA process? What could go wrong and right as we try to quickly make vaccines available to hundreds of millions of people? What does it mean in terms of getting, quote, back to normal? Those are just among a few of the areas that we will cover. And on therapeutics, what does the recent FDA emergency use authorization for Lilly's monoclonal antibody mean? How do we interpret the confusing results on remdesivir? Are there any promising treatments that are emerging as the hundreds, maybe even thousands of clinical trials that were set in motion 6 to 10 months ago, all are coming to fruition? I'm really thrilled that we have two world-class speakers to walk us through these issues whose outcomes are central not only to our health and healthcare system, but to our economy, to our schools, and literally to our future. The second speaker I will introduce first, and we will get to Annie in probably about 40 minutes. It's our own Annie Lutkemeyer, who's been our go-to person on therapeutics since the start of the pandemic. Annie, as you know, is professor of medicine in the Division of HIV, Infectious Disease, and Global Medicine at Zuckerberg San Francisco General Hospital, where she directs the HIV clinical trials group. Prior to all of this, her interests were in HIV and tuberculosis co-infection, development of novel tuberculosis diagnostics, but I think, like most of us, has spent the last 10 months thinking hard and trying to make progress in COVID, and she's been a key campus and really national leader in thinking about therapeutics. So Annie, thank you. We will get to you at about 12:40. We will start with vaccines. And on that topic, I'm honored that we have Paul Offit joining us today. Paul is one of the foremost experts on vaccines in the world. He is the Maurice Hillman Professor of Vaccinology at the Children's Hospital of Philadelphia and a professor at Penn, where he directs Penn's Vaccine Education Center. He was a member of the Advisory Committee on Immunization Practices for the CDC. He is actually a co-inventor of a vaccine for rotavirus, a founding advisory board member of the Autism Science Foundation and the Foundation for Vaccine Research. And he's co-editor of the foremost text on vaccines, which is aptly named Vaccines. He's also an elected member of the National Academy of Medicine. So Paul, thanks so much for joining us. The way we're going to do this is Paul will speak, I think, without slides for the first 10 to 15 minutes, and then we should have about a similar amount of time, 15 to 20 minutes, for Q&A, some of which we'll get from you and some of which I will have myself. So with that, let me turn the stage over to Paul. Thanks so much.
Okay, thanks. So I'll just sort of give a kind of like a 10+ minute summary on where things stand. If you had asked, I think, 100 scientists in the United States a year ago in January of this year, whether we thought we would be where we are now, I think no one would say yes. I mean, we just had the virus in hand. We just had the genetic sequence of SARS-CoV-2 in hand in January of 2020. Now we have two large placebo-controlled trials, one 44,000 done by Pfizer, another roughly 30,000 done by Moderna, looking at a messenger RNA vaccine. So this is a novel strategy. You basically just take the messenger RNA that codes for SARS-CoV-2 spike protein, you put it into a complex lipid delivery system for the purpose of stabilizing it. You then inoculate it into the muscle. That whole construct is taken up by the cell. The lipid construct sort of melts away, and then the mRNA through the ribosomal system is translated to SARS-CoV-2 spike protein, which is then excreted. And then you make an antibody to that protein. So it's a novel strategy. I mean, we ask the body to make the SARS-CoV-2 spike protein, and we may then induce an immune response. The two now right now we're just dealing, as always has been true, it seems like for the last year, science by press release. You hear what the company wants you to hear first, and then you get to look at the data later. But what you've seen so far is that the vaccines are like protective in the mid 90% range, including for people who are over 65, including protection against serious disease. So it looks like we can protect against disease based on these preliminary data. Or I should say top of line data that are put out by the pharmaceutical companies. We will get a look at those data soon. So the way this works now is that the Pfizer has already submitted to the Food and Drug Administration for permission to distribute this vaccine through something called emergency use authorization. Now, when people hear emergency use authorization, they're thinking hydroxychloroquine or convalescent plasma. Where hydroxychloroquine was distributed, and you'll probably talk about this later, was distributed for the treatment of COVID-19, when in fact subsequently it was shown neither to treat nor prevent disease, and in fact have a serious a side effect that included cardiac toxicity. Similarly, with great fanfare, the administration stood up and talked about what a breakthrough convalescent plasma was going to be, even though at the time there were no data suggesting that it or proving that it worked to treat this disease. And so people are skittish. They're skittish about this administration's now approving a vaccine through emergency use authorization. But the difference between hydroxychloroquine and convalescent plasma and these two vaccines is these vaccines were subjected to the size of a clinical trial, as would be typical for any vaccine. I mean, the human papillomavirus vaccine was a 30,000 person trial. The vaccine we developed at Children's Hospital of Philadelphia was a 70,000 infant study. And Johnson & Johnson's is a 60,000 person study now for COVID-19 vaccines. AstraZeneca also has a huge clinical trial. So if that's true, if we're proving that these vaccines are safe, or at least in tens of thousands of people, and proving that they're effective at least for some period of time, why aren't we just doing what we always do? Get a biological license application and get a license, and then go to the ACIP for recommendation. Why not do that? And the answer is because no vaccine would be approved by the FDA that had been tested for this short a period of time. I mean, both the Moderna and Pfizer trials began the end of July. We now are submitting for Pfizer's already submitted, Moderna's 2 weeks away, or a couple weeks away from submitting. So you're only going to have a couple months of data saying whether these vaccines are effective. So you don't know whether they're effective for 6 months or a year or 2 years. You don't know that. And that's the difference. So then the question becomes, have we mitigated a critical amount of uncertainty? I mean, if we do it this way, how much of a risk are we taking? And I think the answer is not a big one. I think if a vaccine is effective for a couple months, it's likely it'll be effective for longer. And although it may not be 90 or 95% effective 6 months, a year from now, I still imagine it would be highly effective. We'll know that afterwards. And the fact of the matter is, you're not going to do a 2 or 3 year study for SARS-CoV-2 when 250,000 people died this year. You're going to live with that degree of uncertainty. Regarding safety, the FDA has insisted that these companies look at least 2 months after the second dose. These are both mRNA vaccines or 2-dose vaccines. Look at least 2 months after the second dose to answer the question, is there any serious adverse event? And so the question then becomes, is that enough time? And I think the answer to that question is yes. I mean, if you look at all the serious side effects that were associated with vaccine, for example, polio associated with the rural polio vaccine, which is rare, roughly 1.2.4 million doses, but real, or say Guillain-Barré syndrome following flu vaccine, which is occasionally happens as frequently as 1 per million recipients, or viscerotropic disease following yellow fever vaccine, or narcolepsy following squalene adjuvanted flu vaccine, or thrombocytopenia following measles-containing vaccine, or allergies associated with latex or gelatin. Those all really occur within 6 weeks. So I can't think. I was on a conference call the other day with CDC. We couldn't think of a serious side effect, truly serious side effect, that really wouldn't be picked up fairly early. There is one thing that worries people, which is so-called vaccine-induced immune enhancement, which is to say that when you get the vaccine and then you're exposed to the wild type virus, that you would do worse than if you weren't ever exposed. There's a lot of reasons to believe that won't happen here. I'm not going to go through them now, but that's one thing people will be looking at. In any case, I think we will have, we'll see what the data show. I'm actually on the FDA's Vaccine Advisory Committee, so I'll be part of the team that votes for whether or not we want to approve it and then send it on to the ACIP. But I think if you had to predict the way this is going to play out over the next few weeks, right now the FDA's Vaccine Advisory Committee has been asked to hold December 8th, 9th, and 10th for discussions about COVID-19 vaccines. I'm not sure whether we're going to be discussing Pfizer's and Moderna's. We'll see. The ACIP has already said that within 24 or 48 hours, they will independently review data and come up with their own recommendations. And then in theory, as early in theory as the third week of December, you could start to see these vaccines rolling off the assembly line into the arms of the American public. So that's my brief 10-minute description. And I did stay within my time, I'd like to point out, and I'm ready for your question.
Thank you. I warned Paul before that it's not unprecedented for 10-minute talks to go half hour. So I wanted to be sure we had plenty of time for discussion, because I think we'll probably hit on all the other areas that you would have talked about. So let's talk about the FDA. First of all, thank you. That was great. FDA approval process. There were a lot of us that were very worried after hydroxychloroquine and after convalescent plasma that the process had been contaminated by politics. It appears to have gotten back on the rails. First of all, do you believe that's so? And second of all, how did that happen?
Yes, I believe it's back on the rails. I think it's back on the rails because Commissioner Hahn finally stood up for the FDA. I mean, I think you're right. I think that if you watch the way the hydroxychloroquine and convalescent plasma played out, especially if you watch that press conference associated with convalescent plasma, you couldn't help but believe that Commissioner Hahn felt that the job of the FDA was to serve at the will of the administration, instead of what its real job is, which is to stand between the American public and pharmaceutical companies to make sure we don't get products that are unsafe or ineffective. In fact, we had just approved 2 products that had not been shown to be effective. So I think I can understand where people are hesitant, but I think what the breaking point came when Commissioner Hahn insisted that there be at least 2 months of safety data following those 2. Before they would even consider approving this through an EUA. The minute he said that, that meant that there was not going to be a submission for the vaccine before the election day. And I think that although the administration pushed back on that, but Hahn stood tough. And I think that was the moment, the breaking moment when he stood up for his agency, because believe me, you want to trust the FDA. I mean, there's an historian named Michael Harris who said correctly, the history of drug regulation in the United States is built on tombstones. It's true. Every major drug regulation is as a consequence of a massive pharmaceutical company disaster. So I give him credit for that. What worries me is that there are now 5 states that said they're going to have their own vaccine advisory committees because they don't trust the FDA. You have one National Medical Association that said they're going to form their own vaccine advisory committee. You can't really have that kind of splintering. It's too chaotic. I do think you can trust the FDA and trust the CDC, no matter how much this administration has disparaged them in the past.
So I think California is one of those states. And I guess, would you recommend they take that down, that they were sort of putting those things up prophylactically because they were worried about the FDA, but there's enough evidence now the FDA is going to do right that they don't need that?
That's what I would say.
Yeah.
Yes, I would definitely say.
Should say for UCSF, particularly medicine residents, Steve Hahn was a former medicine resident here. He was actually co-chief resident with me 100 years ago. And I'm now proud of him. I'm not sure I would have said that 2 months ago, but I think he did the right thing. You talked about the lack of concern about a long-term side effect emerging after the 2 months. How about mRNA vaccine? The fact that it's a new way of doing vaccines And a colleague of mine wrote me the other day and said, is there any concern that these things are going to continue to churn out proteins that are going to cause a low-level inflammatory response, or just something weird we haven't seen before because this is a brand new vaccine technology?
Well, it is a brand new technology. So I think and therefore there are unknowns. And I think you have to be humble and keep your eyes open and realize that there may be things over the next year or two that we hadn't anticipated that happens. I mean, I came into the research world studying rotavirus. I mean, by the time the first rotavirus vaccine came out, which was RotaShield, that was made by NIH in collaboration with Wyeth Laboratories, that virus had been studied in experimental animals and people for 50 years. And when it came out, it was found to be a rare cause of intussusception, intestinal blockage. That was not anticipated by animal model studies. It was not clear at all that natural human rotaviruses caused intussusception. And then there was that surprise after 50 years of study. So I think you have to be humble here that there may be things that you don't know. I do think there are systems in place like the Vaccine Adverse Events Reporting System, the Vaccine Safety Data Link, the vSafe system, the CISA system that is open, that it's opening its doors the minute this vaccine rolls out to see whether there's a problem. I mean, the kinds of things people, remember, you make messenger RNA in your body all the time. You're making proteins all the time. It's a very labile molecule. It tends to break down very quickly. So I don't really worry about that. What people will say to me is, yeah, but won't it incorporate itself into your DNA and, cause you to have a variety of negative consequences? Well, no, because it's first of all, it's messenger RNA, not DNA. Secondly, it only enters the cytoplasm. It doesn't enter the nucleus. I mean, there are DNA vaccines. In order to get the DNA vaccine to get into the nucleus where it needs to go, you have to use an electroporation device, which is essentially an electric shock, which makes the cell more porous, which allows the DNA to get in there. So no, although I never understand why it is that people are always worried that you're going to change your DNA to become, like to have autism or diabetes or multiple sclerosis. Nobody ever thinks that it could change your DNA to become like Spider-Man or have X-ray vision. Why could there be upside to this? But because both are as likely, actually.
You're a glass is half full person. Talk us through the long-term efficacy and immunity issues. So there was a paper, I think, that just recently came out that sort of hinted that immunity may last for a long time. It's hard to know given how little time there has been. How do you read the signals so far in terms of, is this likely to last 6 months or a year and need a yearly booster, likely to last 5 or 10 years? Is there any way to figure that out as opposed to just waiting years and seeing how it goes?
Well, yeah, that's how you figure it out. You wait years and see how it goes. But I think there are some promising signs. The best sign for making a vaccine is that natural infection protects against disease associated with reinfection. That's what you want to see. This virus has been out for about a year. There aren't a lot of people who say that they had moderate to severe disease, recovered completely, and then got moderate to severe disease again. That's good. And so we've had this not for a year now. Two is that people focus always on the longevity of neutralizing antibodies in the circulation, but I'm not so sure that's key. I think the key is and this is true for a number of viruses, is the frequency of memory B and T cells. This is not a short incubation period disease. I mean, and even for short incubation period diseases like influenza or rotavirus, usually after natural infection, you generate high enough frequencies or immunization for rotavirus, frequencies of memory B and T cells. So when you're exposed to the virus, it takes about 3 to 5 days to activate and differentiate memory B cells, say, to become antibody cells, which then modifies infection. This infection has an incubation period of about 6 days, which is longer. So I do think that if there's any reasonable frequency of memory B and T cells, you probably can be highly protective against moderate to severe disease. I think that's true. Again, we'll see, but I'm optimistic.
And let's say it only lasts for a year. How does that spool out? Does that mean you get a booster every year? Is it the same vaccine, or does that imply we've got to invent some new thing to be a booster? Is it, when you need boosters, what is that about?
Well, so it depends what happens to the virus. If the virus is still raging, yes, we would need a booster. And in terms of whether you can boost with the same vaccine or a different vaccine, depends on the vaccine. So I think, if you've gotten, say, a replication defective adenovirus type 26, and then, you're looking to boost with a replication defective simian adenovirus or another different adenovirus 5, it may be difficult to do that. So I don't know. I mean, I think for the most part, when you got sort of Zostavax in 2011, which is a shingles vaccine, and then that was basically replaced by Shingrix. Those didn't interfere with each other. So as a general rule, they don't interfere. I wouldn't imagine they would likely interfere. Maybe one ad vector would interfere with another ad vector, but I mean, the good news is, at least to date, it appears to be a single serotype virus. It doesn't seem to be mutating away from the vaccine as, say, flu does every year.
Right. I was going to ask that. So is there a possibility that we'll need a different formulation every year as the vaccine gets smarter, or as the virus gets smarter? And you think, from what we've seen so far, no?
Well, again, I'm happy to make any prediction you want as long as you don't hold me to it. But I do think that to date, there's no evidence that it's more than a one serotype virus. So hopefully it'll say that.
And people talk about vaccines being better than natural immunity. Are they? And if so, why?
Yes, and for different reasons. I mean, so the tetanus vaccine is better than your immunity following tetanus vaccine is better than your immunity following natural infection with tetanus. The reason is that tetaноspasm, the protein, the toxin that's made by tetanus, can cause symptoms at a subimmunogenic level. That's why. So therefore, the vaccine is better. Human papillomavirus is a better vaccine, actually induces higher titer and in many ways longer lasting immunity than the natural infection does. HPV, human papillomavirus does. Those virus-like particles are just excellent at inducing high levels of immunity. And then when you conjugate polysaccharides like Hib, pneumococcus, meningococcus, babies, children less than 2 years of age don't make very good T-cell independent B-cell responses, which is what a polysaccharide response is. Once you conjugate it, you switch that T-cell independent B-cell response to a T-cell dependent B-cell response. So there are examples where the vaccine's better. The one way it may be better here is that there is some evidence. There were a couple of papers in Science recently showing that this virus actually has the capacity to make you, especially men, make antibodies against interferon, type 1 interferon, which is a part of your immune system that helps work against viruses. It may be that the vaccines wouldn't do that. And so they may be better than natural vaccines. There is some immunosuppressive element to this virus.
Take us through sort of what vaccines do and what we know now about what this vaccine does in terms of preventing cases versus preventing severe cases versus preventing infection and contagiousness, because those are all different ways of looking at it, right?
Right. So I think, again, preliminary data based on press releases, we'll see. But it does look like that the vaccines can prevent disease, meaning mild disease. And then in the case of Moderna's product, there were 11 cases of severe disease in the placebo group. So it looks like it prevented severe disease well. So the bigger question, though, is does it prevent asymptomatic infection? If it prevents disease, great. But if you've gotten the vaccine, can you be comfortable that you're not still going to become an asymptomatic shedder, which is going to infect other people? So none of these vaccine trials are designed to detect that. But there is an interest in, Larry Corey, actually, at University of Washington, is interested in now setting up a trial in college students to answer the question, do you prevent asymptomatic shedding? Because if you don't, or you don't do it well, then you really should wear a mask, even after you've gotten the vaccine. And I think that it would be nice to have data to show that.
So, yeah, take us through a little bit about the herd immunity thing, because you really should wear a mask, but what if 70 or 75% of the population truly is protected against getting sick from the vaccine? How do those two things play off against each other? It's a little confusing.
You should probably wear a mask anyway. But let's say it is 75% effective. You may be that 1 in 4 people who isn't protected. So therefore you would benefit from wearing a mask. And the mask is the smartest thing we can do. It's frankly the lowest tech thing we do, and it seems to be the thing we're worst at. I mean, we just spent $24 billion making 2 vaccines that look amazing in a year, and yet we can't bring ourselves to wear masks and social distance, which is the lower tech solution. And it's also very powerful. I mean, you can really stop spread if you do this right. We just somehow think it's our freedom to catch and transmit potentially fatal infections by not.
Yeah, for sure. I mean, I hope everybody on the call knows that and is doing that. I guess I'm asking about, fall next year. Let's say, and we'll talk about sort of how many people will take the vaccine and all that kind of stuff. But let's say 75% people end up taking and getting, there's enough supply to get a 95% effective vaccine. Do you envision that, and you could argue we should have all been wearing masks for the last 100 years for flu anyway, and we don't. So what do you envision the state of the world will be if you have that level of vaccination rate and you've reached a number of vaccinated people that at least most people say is the herd immunity threshold? Do you still think that the risk is high enough that we would have to not live our normal lives in terms of crowds, or or mass, or do you think we get back to something resembling normal?
So there's actually a formula for what percentage of the population you would need to immunize based on what the contagiousness of the virus is and based on what the efficacy of the vaccine is. But if you had a vaccine that was, let's say, 90% effective, if you plug it into the formula, assuming an R naught of 2, and let's just make that assumption, then you would need to vaccinate about 55 to 60% of the population. But that assumes, say, 90% effectiveness against at least important shedding. You still may shed to some extent, but the question is, are you shedding at a level that's contagious? So it's not necessarily just plus minus, and that needs to be worked out. And there are other things obviously working against transmission, which is hygienic measures, and awful as it is, I mean, there are people who've gotten this infection and survived it who probably are at lower risk now of getting infected. And that could be probably in the vicinity of 40 to 50 million people, because we clearly underestimate the number of cases that have happened.
Mm-hmm. Okay. Should people get antibody tested before they get vaccinated?
No.
How come?
Let me make this easier. It's hard to get a vaccination program in place anyway. One of the companies, I think it's Pfizer, has excluded people who were antibody positive. Moderna didn't. So there's going to be data on people who were infected, who had antibodies, who got the vaccine. So you'll know about those data. But I don't think there is any interest in trying to screen people out first. Because first of all, the antibody test you get doesn't necessarily tell you you're protected, because we don't know what that antibody test is yet. And I don't see any downside other than boosting. So
okay.
Upside.
So, but even if it's logistically too hard to have that be part of a program, if you knew for sure you had positive antibody, you had COVID in March and you have positive antibodies, you would still say, just get the vaccine. It's the right thing to do.
Yes.
Okay. Let's talk about distribution. So we're trying to distribute, vaccines to 330 million Americans and billions of people around the world. And we have freezer requirements and tracking requirements of making sure you get the same one twice. So it's sort of like, what could go wrong? Well, tell us, how is this going to go? What could go wrong? What needs to go right in order to make this work, and kind of what that implies in terms of the timeline?
So I think that I've been working with our Secretary of Health here in the Commonwealth, Rachel Levine. I'm part of a team that she put together. And what I've watched her just be great at this, what she has done, and she, our Commonwealth has very, large urban areas like Pittsburgh and Philadelphia. And then we also have very sparsely populated rural areas in the center of the state, Commonwealth. And so she has really done a good job at trying to essentially set up centers, the vaccination centers, because she's concerned that this could really work at the level of retail pharmacies because of the Pfizer's requirements for shipment, storage, and handling, which are hard. And so she's got all that down. She knows what she wants to do. She knows where to do it. She's got buy-in from the people in the Commonwealth. What she needs is all she needs from the federal government is, let me know how much vaccine I'm getting and when, and where you're shipping it. And 2, I am going to need some help financially to set these things up. That's her attitude. You don't have to tell me what to do, just to do that. And I think that's where the rubber is going to meet the road on all this.
And are you, not, as we've seen in the response so far, not every state thinks about these issues the same way, is equally competent, has the same leadership. So it sounds like you're talking about a state by state process where what the feds do is just, FedEx envelope shows up and here's 1 billion doses of vaccines, and it's up to the state to distribute. Is that the right call, or should this be managed more centrally?
Well, we're about to have a new administration. So I think when the Biden-Harris administration takes over on January 20th, there may be clearer directives from the federal government about how this is going to play out. Obviously, right now we have none. We have no federal leadership in this.
Yeah. The freezer requirement for the Pfizer vaccine, how hard is that to deal with? How much does that make you prefer the Moderna, or not so much?
Well, first of all, so here's how hard it is. It has to be shipped and stored at -70 to -80 degrees centigrade, which means you have to maintain it on dry ice. We've never done that for a vaccine this in this country. You have to have the right kind of storage containers to ship and also to store in your facility. Once thawed, you only have a day in the refrigerator before you have to throw it out. So it only has a shelf life of 1 day in the refrigerator. It also comes in 5 and 10 multi-dose vials as a concentrated product. So it has to be lyophilized. It has to be reconstituted with just physiological saline. Once reconstituted, it has a shelf life of 6 hours. So that's hard. The good news is BioNTech, which is the inventor of this vaccine that's working with Pfizer, it sounds like they're going to be able to create a complex lipid delivery formulation that mimics Moderna's, so that they'll then have the same properties. I mean, there's no difference between the two molecules, not functionally. They both really express the same receptor binding domain for the spike protein. So the molecules are, so I think, equally labile. It's that complex lipid delivery system, Moderna's versus Pfizer's, is not the same. But I predict within a year, I think that Pfizer will figure this out.
And these are not the only 2 vaccines that are being trialed. We heard some good preliminary news about the Oxford one today. So what's your level of confidence that others will join the chorus here? And how important is that? We have 2, is that enough, or we need more?
No, I think that to vaccinate, as you said, sort of 300 million Americans, we definitely need a few vaccines. In terms of how those vaccines apply, the AstraZeneca product is a replication effective simian adenovirus. The Johnson & Johnson product is a replication effective Ad26, with which we have at least some commercial experience. That was the Ebola vaccine in West Africa. So we'll see. I mean, I'm open-minded. These are sort of novel strategies, the so-called genetic plug and play strategies. The reason that they were done, as you said, I think at the top of this, is that they're the easiest to construct, and they're very easy to also mass produce. That's why they're first, not necessarily because they're best. And the history of vaccine development invariably is that the first vaccines are not always or often the last best vaccine. So you just want it initially to be good enough.
Well, it's hard to imagine that they could be better at 95%, but is it likely, because we've discovered that the spike protein was the right target, is it likely that other vaccines will be as efficacious as these were?
Yeah, I would be cautious about saying these vaccines are 95% effective. Really? I think that what I would say is that right now, 1 week after dose 2, we show that there's 95% efficacy against disease. That's great. There's some things that worry me a little bit. I'd like to, they in theory have 2-month data after dose 2, because that's required for the safety issue. So if they have 2-month data after dose 2, why don't we see those data? Why was it just 7 days after dose 2? Why just a week after dose 2? We have the 2-month data. Was there any erosion of efficacy, which may well happen? 2, if you look at the placebo group, the incidence of disease was lower than you would expect, given what the incidence of disease was in the area. Why is that? And one reason you could explain that is so-called volunteer bias, that the kind of person that chooses to enter that trial may be someone who is more likely to wear a mask, more likely to social distance, therefore less likely to get infected, or when infected, is infected with a lower inoculum, which would be then easier to protect against, because there's clearly an inoculum effect. The higher the inoculum of virus, the more likely you are to have moderate to severe disease. So I'm really looking forward to our FDA's Vaccine Advisory Committee meetings, which are going to be the 8th, 9th, and 10th. I'm looking forward to asking those questions and having the FDA and the companies explain all of this, so that we can have a much better idea when we say 95% effective, exactly what does that mean?
Yeah. And you're looking forward to reading those 100,000 pages, I'm
Yes,
sure.
Really, 100,000.
Every line. I've heard skepticism, quite understandably, that these things have come out by press release, and these are companies that are trying to make a profit and see their stock price boosted. I've sort of countered that if I was Pfizer, the idea of getting it wrong and that being discovered will have so much brand destructive impact that this is one where you want to play it straight. What's your sense of the way they are putting out information right now?
Yeah, I mean, there's no hiding. If they're making a product that is either ineffective or unsafe or both, it will be found out. I mean, this is the thing. I mean, you can talk all you want about sort of pharmaceutical company hype. Once the vaccine rolls out, between VAERS, the Vaccine Safety Data Link, post-marketing surveillance in terms of efficacy, you're going to find out the strengths and weaknesses of this vaccine. So if they know something that is, in any case, sheds negative light on their vaccine, it's going to come out.
Yeah. All right, let's turn to one last question about the process so far. Many of us have been critical of the administration on almost everything around COVID. And yet, at least as I have looked at this, it feels like Operation Warp Speed did what it intended to do and did it remarkably well. Am I missing something?
I think, as painful as this is to say, I think the administration should be praised for what, frankly, is roughly a $24 billion effort that within 1 year, and we'll see when we look more closely at the data, has created 2 vaccines that appear to be highly effective, safe, and work well in people who are older. It's a remarkable technological achievement. Amazing. So, I mean, I was quoted actually in CNN, and it was a joke. And I unfortunately they ended up printing it, even though she told me she wasn't going to print. You know, I said, you should praise Tony Fauci and you should praise Peter Marks at the FDA for setting this all up. I said, I don't think it was Donald Trump. I said, who I don't think can tell the difference between a white cell and a prison cell. And they printed that. So meanwhile, I haven't heard the end of it from my hospital yet.
We'll do a little lesson later on talking to the press.
Right.
Yeah. But I've heard you comment elsewhere about one of the things they did that was so important was to de-risk the process for the pharma companies. So talk about that. What did that mean?
Yeah, they took the risk out of it for pharmaceutical companies. They said, look, we'll pay for phase 3 trials, even though that costs hundreds of millions of dollars. We'll pay to mass produce this vaccine at risk, even though that costs hundreds and hundreds of millions of dollars. We're willing to throw away hundreds of millions of doses of vaccine if it's shown not to be safe or ineffective or both. I mean, the pharmaceutical company would never do that. That's what moved this along. It's the same thing as the polio vaccine in the 1950s, where it wasn't the government that did that, it was the March of Dimes that did that. But that's also why we were able to make a polio vaccine so fast in the mid-50s.
Yeah, I think people, some of it was the science, but some of it was the business, that it became the reason it takes so long sometimes is actually the difficulty of this from a business proposition. It sounds like that's a big part of what they did here.
Yeah.
We have a few minutes left. I want to talk about people and their willingness to take the vaccine and the politics of that. So first of all, when you see a Gallup study a month ago that's saying 50% of people won't take it, and then a more recent study, actually before the news though, about Pfizer and Moderna had gone up to 58% of people said they would take it, 42% would not. How do you interpret that?
Well, you're asking them whether they would take a theoretical vaccine. I mean, there is no vaccine yet. I think when we know more about this vaccine, as the details of this vaccine get out there, as people start to get a vaccine, especially essential workers like healthcare workers, transportation, et cetera, and now hundreds of thousands of people have received it, and millions of people have received it, and then let's assume it's still safe, still effective. But then I think the people will be more and more willing to take it. And most of the people who are being asked to get this vaccine are healthy young people who wouldn't normally die from this virus. So it's reasonable to be skeptical, and you don't want to see any sort of serious side effect. Maurice Hilleman, who I think is the father of modern vaccines, said it best. He said, quote, "I never breathe a sigh of relief until the first 3 million doses are out there." That's going to happen pretty soon. And then I think people will feel better about this.
Let's talk about this whole anti-vaxxer thing. And I know you've had to deal with that for probably most of your career. So what are the insights that you have about the world out there that distorts the science or has political agendas that bleed into this? What should we know about that, and how do you think that's likely to play out in the specific case of COVID?
Yeah, so I mean, the true anti-vaxxer, meaning the conspiracy theorist who isn't going to believe a thing that you say, including solid, clear, reproducible data. But they are very good about at least getting their message of hate out there on the internet. Joe Mercola is one perfect example. He's got all this stuff now about sort of antibody-dependent enhancement and vaccine-induced immune pathology, for which there is no evidence. And actually, there's a lot of evidence against that. So yeah, you're going to be dealing with that. I mean, I'm on the NIH Active Group, which was put together by Francis Collins, and we were asked to really address the issue of safety, which now we've written up and is going to be a Science Translational Medicine paper. But I also worked with the group to just come up with like a 3-pager that sort of simply describes that for the public. So I think our job as clinicians and educators is to try and get good information out there and just keep getting it out there and getting it out there and hope for the best. But they're never going to go away. But I do think that this vaccine, if it is what it looks like, it's going to be, I think the vaccine will be the hero of this story.
Yeah. Do you, I guess, worry in the world of social media and different information sources? There will be cases of people getting the vaccine and having a stroke, and getting the vaccine, having a heart attack, getting the vaccine, developing cancer, because that's called statistics. It will happen. And that can be distorted. Anything that we can do to prophylax against that, or you just think that's coming?
No, I think what we have to do is, because by definition, I mean, this vaccine is going to be designed to prevent SARS-CoV-2 infection, not everything else that happens in life. So there are going to, especially when you're vaccinating an older population who are going to have medical problems, people are going to get a vaccine and then have stroke, heart attack, whatever. So you have to have in place, and I think with the vaccine safety data link, we'll have that. Look here, there is no greater, this is occurring at a background rate. It's occurring at the same level in people who haven't gotten the vaccine yet. You have to be able to arm clinicians and educators with that kind of data. And that's what the CDC can do. And I am really looking forward to this next administration so that we can start to resurrect these science-based agencies, which have just, I think, suffered at the hands of the current administration.
Thank you. Maybe my last question. I'm looking through the questions that have come in. Any hope, someone asked, is there any hope for an HIV vaccine with this novel technology? And maybe I'd broaden the question to, and Annie may want to weigh in that later, but I'd broaden the question to, in the things that we have learned in the last year to develop these vaccines in record speed, is there something about that will have enduring effects as we approach other infectious threats?
We'll see. I mean, it's interesting, isn't it? I mean, people have brought up not just for HIV vaccine, but as a universal flu vaccine, malaria vaccine. Is there anything else we can get from these novel strategies that'll help us down the line? I think we'll see. It certainly opened the door, I think. So it'll be interesting to see how this plays out. I mean, I certainly hope there's not a serious adverse event associated with these vaccines. It'll be a huge setback, but we'll see.
All right, Paul, one personal question. So I'm on the wards starting December 20th, which now, and UCSF is a vaccine distribution site. They offer me dose 1 on December 15th. Are you recommending I take it?
Yes. Yes. Well, again, let's see what the data show. I mean, assuming the FDA and the ACIP approve this vaccine, then they will have carefully looked at the data. And if they say someone, your age, should get this vaccine, I would say,
I get it. Yeah.
People over 45, people like you.
Yeah. Yeah. Well, I, from what I've heard and from what you've told us today, I think I will be first in line. So thank you for spending time with us, and thank you for your leadership in this area. It's been really incredibly important, and it's extraordinarily exciting where we find ourselves. So hopefully 2021 will be a much, much better year because of this. Thanks so much.
Sure. Take care.
All right. Let's turn over to Annie. Annie Luetkemeyer, who's a professor at Zuckerberg San Francisco General, will take us through what we should know about some of the new developments in the world of therapeutics. So Annie, it's yours.
Terrific. Thank you so much. So I'm going to go over 2 and possibly 3 things depending on the amount of time. And this really is a glass half empty, half full update. We're going to talk about monoclonal antibodies, which have both half full and half empty, and then remdesivir, which is on the right-hand side of the screen, which is an RNA polymerase inhibitor, which has engendered some controversy. That seems to be the theme of 2020, even in medicine. So let's just dive in. Next slide. These are my disclosures. Next slide. So monoclonal antibodies. Let's just take a step back and talk about what they are, and then we can get into the data a little bit. So monoclonal antibodies are laboratory produced antibodies with a single COVID target, and they typically target a spike protein of the coronavirus. If you give several monoclonal antibodies together, they're combined as a cocktail, but each one targets a single part of the coronavirus. And the way that these are developed is that you take blood from someone who's recovered from COVID-19, you figure out what the best targets are, what is adhering the best, which antibodies have the highest affinity for the spike protein, and then you put that DNA into cells that then mass produce these antibodies. This is not new technology. We've used monoclonal antibodies for autoimmune diseases, for cancer, for lipid lowering, but it is new in the coronavirus field. They are given IV or subcutaneously if you can get the volume down. Right now, most of them are being looked at in IV, but certainly would be much better to go subcutaneous. The good news is that they have a long half-life, so weeks to months, which makes a single dose possible. Coming in to get an IV infusion, particularly if you're infected with coronavirus, is challenging. And they are being evaluated across the spectrum of COVID disease, inpatient, outpatient, and then very excitingly for use in prevention. They're generally well tolerated. There's a rare risk of allergic reaction, and for this reason, people need to be monitored after they are given these vaccines. Given these, I see a vaccine on the brain. Given this rare risk of allergic reaction, but in general can be given to people with liver, kidney problems without an issue. Next slide. So I'm going to start with the glass half empty here. These are two of the monoclonal antibodies that are furthest down the road, but there are many, many under evaluation. So first, talking about the Lilly CoV555 antibody, now easily named bamlanivimab. I'm sure that I botched that. So this is a single target antibody that was in the ACTIV-3 study, which is a study looking at hospitalized patients. And we got the news several weeks ago that this arm of ACTIV-3 was halted for lack of efficacy. We were told there was no safety signal and that the study will reopen, including other monoclonal antibodies shortly. But I think certainly viewing this as a setback, at least for people who are already critically ill enough that they require hospitalization. Shortly thereafter, we also received the news about the Regeneron CoV2 combination. This is a dual monoclonal antibody combination, similarly being studied in hospitalized patients, so people who are already sick enough to need to be in the hospital. The DSMB there found that they recommended putting the study on hold in only a subset, so in ICU patients and in those who are on high flow oxygenation. And again, Science by press release, the information that we have is that it was stopped due to potential safety signal as well as unfavorable risk-benefit profile, but will be continued in those who are hospitalized with low or no oxygen requirement. So why did this happen? I think we obviously need a lot more data to understand it. Remember that monoclonal antibodies act basically as an antiviral to really drive down the amount of virus in a very quick way that is replicating in the body. And so I think that the leading theory here is that once people are hospitalized, the horse may be out of the barn, and that an antiviral approach, while good, may need additional bolstering from anti-inflammatories once people are critically ill, or for something like a monoclonal antibody, perhaps it is just too late. So we don't know, but I think that this was worrisome, particularly when you see two separate agents coming to the same conclusion in a similar study population. And in this case, this was critically ill patients or severely ill patients who are in the hospital. So that's the glass half empty. Next slide. Well, what about in outpatients? Do we still have some optimism here? And I think that there is some optimism. We saw a recent publication in the New England Journal looking at bamlanivimab versus placebo. So this is the single target 555 Lilly antibody. And this was given within 3 days of a COVID positive test to outpatients who had symptomatic COVID disease. And that's a pretty high bar to get people in very quickly after being diagnosed and then linked to care. It was a little over 450 patients, and they looked at a number of different endpoints, including reduction in viral load. And you can see in the upper right-hand corner that there was a reduction in viral load in one of the doses, 2,800 milligrams. But I think importantly, if you look at all 4 arms, including the placebo, you see what you would see in the natural history of this disease, which is that the viral load tends to come down. So how much more is it being driven down by having the monoclonal antibody? Well, maybe a little bit more in one of these arms. Is that where the action is? We don't really know. So we need to understand, it would be nice if this was statistically significant in all of the arms and there was a very big signal, but we know that this is already the direction that patients are going in when they are recovering from COVID. So it's a little bit of a challenging primary endpoint. Well, what about the other endpoints that we care about? What about staying out of the hospital or the emergency room? And unfortunately, in this paper, they combined hospitalization and emergency room, which I think are different in my mind, but these were combined as an endpoint. And what they found is that overall, there was what looked like a statistically significant reduction. Only 1.6% of people were hospitalized if they'd gotten an antibody versus 6.3% overall. So that's a pretty big reduction. But as you can see, this is a very small number of people who needed to be hospitalized or go to the ER. Well, what if you look at those who are at the highest risk? And here they categorize them as those with over the age of 65 or a BMI of 35 or greater. And this was a post hoc analysis. And they saw that this difference was a bit larger, where 4.2% were hospitalized with the antibody and 14.6% were hospitalized if they got the placebo. So again, I think a nice signal that we're seeing there. And what about symptoms? How did people do in terms of feeling better? Well, it certainly looked like it was going in the right direction when people received the antibody versus the placebo. Also, there are data from Regeneron, which I'm not going to have time to go through today, and I included the press release on the same day, that also reiterate this trend towards a decrease in viral load, a trend towards a decrease in hospitalization. And interestingly, in the Regeneron data, they really focused on people who were antibody negative at the time of administration, and they found that those are the individuals who responded best. We don't have that data for the Lilly antibodies, but we hope to get that, at least I hope to get it when we get more data on these analyses. So what does this tell us? I think this is a good proof of principle that outpatients may be different from inpatients in terms of the efficacy and the potential for this intervention. But I think that's really what these data are. They're a proof of principle. They're not overwhelmingly definitive that this is a done deal and that we should be using these monoclonals in all outpatients. Next slide. I think that the FDA approval of the Lilly 555 reflects this. This is from last week. This was the FDA approval for the Lilly antibody, and they applied, I think, some appropriate restrictions here, although I think there is some debate and controversy about whether this was still a little bit too soon. So in the EUA, they limit it to higher risk patients, and I included in the box that you can see that this is over age 65 or with some worrisome comorbidities, same over the age of 55, but have to have cardiovascular, hypertension, or COPD. So this is not for all comers. We need more data for people who are 30 and outpatients and who are feeling sick and don't have comorbidities. There is a short window for administration. It's limited to those within 10 days of symptom onset. And again, while that sounds like a lot of time, it's really a difficult, I can tell you from someone doing some of these studies, it can be a heavy lift to identify people, link them to care, and get them into a medical center to infuse them. It's a 1-hour IV infusion with 2 hours of monitoring afterwards, and this is in the outpatient setting only. This is something we can do relatively easy in the inpatient setting, but it's not permitted in the inpatient setting for the data that I showed you. So then you have to think through, how am I going to give this in the emergency room or in an outpatient respiratory care clinic to a person who's within the infectious window, and it just raises some logistic challenges. Supplies are going to be limited, and this is being allocated through HHS. So this is not something that is, there's a huge supply available. We don't know what the magnitude of the clinical benefit is, and I think we need to better understand the role of baseline antibody status. I included the equipoise expressed by the NIH COVID guidelines here, where they said there's insufficient evidence for or against use of monoclonal antibodies despite the EUA. They stated that they thought it was not standard of care, but that it could be considered for those at highest risk. And I agree with this. I think this is a step forward for outpatients, but right now with the limited data that we have and the supply constraints, I think this is unlikely to be a big game changer, but I would consider it for patients who are at highest risk, who are outpatients, if you have the systems in place to administer it. But I think we are all looking forward to more data, and encouragingly, many of these studies are still ongoing if they've been shown safe to proceed. Next slide. So I'm going to shift gears to what the heck is going on with remdesivir, which is one of the other controversies that has been coming up du jour. So on the left-hand side, I'm showing the NIH treatment guidelines, which nicely lay out what the recommendations are for use of remdesivir. And then this is in contradistinction to a lot of press that we're seeing about a recent WHO study that we're going to discuss, the very bad look of remdesivir published in, as an opinion piece in Science. And then tomorrow, I think we're expecting the WHO will issue conditional guidance about remdesivir as part of their ongoing living guidelines, and they're expected to recommend against remdesivir use. So what is this all about? And to put this in context, what I want to do is to not take Team Remdesivir or Team WHO, but to really just go through what the data tell you, what the data tell us. And I think that there is a way to have both of our sources of data, or all of our sources of data, be incorporated here, because I don't think that they're at odds. Next slide. Okay, so what do we know about remdesivir here? I'm showing you the where it acts on the RNA polymerase. to block replication. So some of the first and most robust data we have come from the ACT-1 study, and this is a randomized controlled double-blinded study that recently had its final report come out, but this was reported much earlier in the epidemic, as well as a preliminary report. And just to remind us what we do know from this study, and this was over 1,000 people who were randomized. The primary endpoint was time to clinical improvement, and this was statistically significant, so 15 down to 10 days. 8 days less of receiving oxygen, lower progression to non-invasive and invasive ventilation, and a significant mortality benefit in those in low flow oxygen. It's hard to lump mortality all together because there's a very big difference between someone who's in the ICU and someone who's on no oxygen. So it's important to look at what the primary endpoints are and have caution about subgroup analyses, but also just to recognize that these groups are very different, and when you have the power to do so, it is worth looking at these subgroups. Generally safe, less adverse events than placebo, and importantly, a better response if given within 10 days of symptom onset, but still did look effective in those who received it more than 10 days after being treated. And just a reminder here, looking at the baseline status by the ordinal score, that people had the best response if they were receiving oxygen, and this is also where we saw the statistically significant mortality benefit. Next slide. So where is the controversy coming from? So there was another large study called the Solidarity Study. It's a pragmatic, multi-center, open-label trial that really should be commended for taking on this heavy lift. They have 11,000 people enrolled in more than 30 countries all around the world and generated some terrific data under challenging circumstances. In the remdesivir component of the study, 2,700 were randomized versus the control, which is standard of care. And again, as a pragmatic trial, they weren't able to have as much granular level detail, and they really focused on mortality, ventilation, and length of stay. And they didn't see a benefit of remdesivir overall in all comers on mortality, the need for ventilation, or the length of stay. And I'm showing the Kaplan-Meier curve in the upper right-hand corner. So next slide. As part of the preprint publication from which these data come, they did a little bit of a meta-analysis looking at the mortality from in the Solidarity study, and in the red box, you can see that when they parse it out by no oxygen, lower high flow oxygen, it looks like there might be a trend there, or ventilation, they were not statistically significant. And just in comparison, you can see that the ACT data do show that in low flow oxygen, there was a statistically significant benefit, but that overall it did not meet statistical significance. So I think that this is a fair evaluation, and we're really happy to have the Solidarity data. They were not positioned to address the same endpoints that ACT addressed, which was time to clinical recovery. So next slide. I want to just put this then into context of what I think we know about remdesivir in the context of both of these studies. So is there a clinical benefit? Yes, it does appear to be a clinical benefit that was well demonstrated from ACT-1. That doesn't mean that the findings from the Solidarity study about mortality are not important. And they did not show a benefit in the Solidarity study for time to ventilation, but we certainly saw a benefit in terms of time to clinical recovery, particularly in some subgroups in ACT-1. Well, so what about the mortality benefit? Yes, it is not seen in all disease stages and when taken as an overall composite. But I think we can't ignore the significant mortality benefit that was seen in those in low flow on ACT-1 with all of the appropriate caveats about subgroup analyses. We have plenty of interventions that we provide in medicine that do not statistically significantly impact mortality, but provide crucial clinical care and really impact people's recovery. And I think that that's important as well. It's safe, so safety is really critical here. But I think it is important, and I anticipate that the WHO guidelines, as I understand it, are going to address things like cost, the need for IV administration, and access globally. And these are important to consider in the US and globally as we allocate resources. But my job and my goal here today is to talk about whether or not remdesivir works. And then we have to sort out from a public health point of view how we choose to get it to others. What we don't know if there might be more benefit in the ICU if patients are followed longer. This was an issue in the ACT-1 study. How late is too late in antiviral therapies? And what is the additive benefit of remdesivir when given with anti-inflammatories? And might there be more of a benefit in those who are critically ill? We know that dexamethasone really showed a remarkable benefit in those who are in the ICU. We really need to understand what happens if we give remdesivir there. What about adding other drugs on top of remdesivir? We're going to see some exciting data coming out very soon about baricitinib. So I just want to close with, I think, a well-said statement from the FDA that came out in the frequently asked questions here, and that basically acknowledged that these data coexist with each other and that they are both, I think, valid and came from studies that were well done and designed to answer different questions, and that the Solidarity results do not refute the findings of benefit to patients that were established in Act 1. Next slide. I referred to a bunch of guideline references here, so I wanted just to include them here. This is the IDSA, the NIH, and then our Division Weekly Digest. I'm going to skip talking about baricitinib, except to jump to one slide. So next slide. Next slide. As a bit of a teaser, these were presented publicly at a recent conference, but should be coming out in a peer-reviewed manuscript very soon. But to address the question of, well, how do we do with remdesivir when we add on immunomodulators? And while there was a modest benefit overall with one-day improvement with baricitinib, which is a JAK-STAT inhibitor that is used in rheumatologic diseases, again, the action really comes when you dig into different disease levels of disease activity. So you see a much bigger impact when you look at those who are on high flow oxygen, where the difference went up to 8 days. And when you start to parse out these data and look at it in comparison to dexamethasone, I think we start to see some very, very interesting findings, which hopefully will be coming your way very soon. So I will stop there so that I can stay on time and make sure that there is time for questions.
Great, thank you, Annie. It's terrific. So a couple questions on monoclonals. What do they do for your overall and long-lasting immune response, and what is the interaction between monoclonals and ultimately vaccination?
Yeah, so it's a great question. So as I said, they have a long half-life, but it's not a interminable half-life. So they should clear the body, and this is not a way to induce long-term immunity. And so even in prevention studies, if you're looking at very vulnerable populations, as a prevention strategy, you would have to repeatedly dose these if there wasn't another way to induce immunity. There's a theoretical concern that if you drop the viral load very quickly with either convalescent plasma or monoclonal antibodies, that you might impair the natural immune response through antibodies or through cellular mediated immunity because you didn't give your own immune system a good enough chance. It's hard for us to really measure that, but I think given that we're trying to use these treatments to treat ill patients, that's a risk that we are willing to take, but we'll have to follow up in the long term to see. We know that people who were less severely ill look like they may have a less robust immune response. And are you then inducing that state? Lastly, how will this impact vaccines? Well, the vaccines right now are not therapeutic vaccines, but let's say in theory someone got vaccinated because they didn't think they had COVID and then got COVID the next day and then came in and wanted to get a monoclonal antibody. Could giving that antibody impact the efficacy of the vaccine in theory? I think in theory, yes, it could, because as Paul really nicely walked us through, these vaccines cause the body to develop the spike protein, and then you're relying on the body's response to that protein. So yes, in theory, I think that would be a pretty small group of people to whom this would apply, but it is an interesting idea.
But I guess the idea, if you had gotten a monoclonal a month ago, and now the vaccine's available, in Walgreens, would that potentially get in the way of your ability to make the spike protein because you already have monoclonal floating around to attack it?
I think you'd probably make it, but it might impair your immune response because it might sort of sop it up, which is the goal of the antibody here. So it would probably be the better part of valor to make sure you're outside of the half-life or well outside of the half-life there. So probably closer to 2 months after getting a monoclonal antibody. All that being said, you had a monoclonal antibody because you were sick in theory with COVID. And so we think that people are actually reasonably well protected within those first 90 days. Reinfection can happen. It's uncommon. So I would say that it's would be absolutely fine for someone to wait because they're probably still within a window where, barring, severe immunocompromise, they're probably protected from reinfection.
And once people are vaccinated, let's look out 6 months from now or so, not only is it preventing cases, but it's preventing severe cases. Does the risk-benefit equation, one of the things that sort of thinking through, when can we go, quote, get back to normal? And it probably relates to both having a vaccine that's highly efficacious, but then people will still get some cases of COVID, having therapeutics that if someone does get infected can mop it up and decrease the risk. Does the fact that the vaccine also seems to decrease the risk of a bad case of COVID change the risk-benefit equation for any of this, for monoclonals, for remdesivir? Do we have to sort of rethink or retrial these things in a post-vaccine era, or will the data still hold? You know what I mean?
No, I hear what you're saying. I think that all of these therapeutics, unfortunately, so far are really focused on severely ill patients. We know we need outpatient therapies, and I would hope monoclonals will have a niche there, but there's going to be some logistic problems. But all the ones that we're really focusing on, steroids, remdesivir, all of these immunomodulators, are really for sick patients. So even if the vaccine works great, if there are people who develop severe disease because the vaccine failed or for whatever reason, I think they're still in front of you with severe disease, and we will need to treat them with the tools that we have available. And I can't imagine that because they've been vaccinated, that would affect how an antiviral or an anti-inflammatory would work. I hope we'll have a whole lot less of them, which will overwhelm our medical system less, and we can do a better job of caring for them. But I think that dexamethasone will work the same whether or not you had received the vaccine and if you were a failure. And I don't think it's going to be 100%. I think we're still going to have people to take care of, although I hope they'll be much less ill.
Colette Dejean asked, any thoughts on why the FDA approved Lilly's EUA for a lower dose and longer time window, 10 days instead of 4 days, than what showed efficacy in the phase 2 trial? Could that potentially waste a scarce resource?
So they approved it at a lower dose at 700 milligrams instead of at the 2,800 milligrams. It looks like in general, there's not a whole lot of dose responsiveness here. And either that, I don't know why that is, but possibly it's because you've already sort of saturated what you need to do. My understanding is that they went then with 700 because they thought it was going to be efficacious and that they did want to help preserve the supply chain. And they went with symptomatic within 10 days based on other data that they have. It was going to be a hard bar to say within just 3 days. So there's not only the New England Journal paper, but also the data that are in the EUA and a bunch of press releases. And so they're triangulated a bit there.
Yeah. And do you envision at San Francisco General that you will be giving monoclonals to people? Are you set up? I'm just trying to think through logistics. The test takes a day or two to come back, and all right, I'm positive, and now I got to figure out where I'm going to get it, and I can't get it in the ER, and I can't get an outpatient clinic because I'm going to infect everybody. So there's got to be some specific setup that's fast track for me to come in and get an IV infusion and sit there for a couple hours. Seems really tricky.
Yeah, I can tell you, having spent a lot of time setting up the ACTIV-2 study, which provides monoclonal antibodies to just this kind of patient, took a lot of logistics to figure out where we could bring in patients to get exactly as you say, identify them quickly enough. We have to get them within 10 days. They're still symptomatic. They're definitely still infectious. Where can we safely infuse them, monitor them? And we ended up setting up a unit that's basically like an ER unit, but that's completely self-contained in our old ER. And so you really have to be thoughtful about how you're going to do this. ERs don't love prolonging the length of stay in the emergency room, particularly with folks with COVID. Respiratory care units, many of them have been able to set up COVID infusion aside from monoclonal antibodies, and so they can add this in, but it is not a small thing to do this. I don't want to be dismissive. Look, if this works well, we need to figure it out for our high-risk patients. Keeping people out of the hospital is a really big deal, and it helps as much as the hospitals are a wonderful place. The more we can keep people out, the better off they do, and we all do. So if it really holds that we should be doing this, we're going to have to be really thoughtful. And I would say the same thing goes with remdesivir. Can you give people 3 doses? But it has created a lot of logistic headaches to sort out exactly how to do this. This is only for 1 dose.
Yep.
But we're working hard to figure that out. And it took a while to figure it out for the research piece.
Yeah, I'll bet. On remdesivir, we only have a minute or two for questions. On remdesivir, you made the point that the Solidarity, and I guess the FDA made the point, Solidarity trial doesn't obviate the results of the prior trial. Did it change your approach at all? Were there patients that you previously would have given remdesivir to that you now don't recommend it, or you still kind of where you were before all this?
Yeah, I mean, I think remdesivir, our practice has been influenced by the shortages that we went through. So I think we all want to be thoughtful stewards of resources, and we had to be very careful for a while because we ran extremely low on remdesivir. So that has to be a consideration. But if patients come into the hospital hypoxic, including critically ill, I give them remdesivir, and then I figure out what additional agents they need. If they really have an high oxygen demand, we'll add dexamethasone on board. I think the area that Solidarity didn't address, but where I think the jury, for me at least, is still out, is that the benefit in people who are in the hospital but only without an oxygen requirement. There's some data to suggest maybe a modest benefit there, but it is, I think, quite limited. So I have not stopped giving remdesivir, and it may be that the mortality benefit in some groups really is quite limited, and I'm okay with that. We all need the mortality to come down. We need better drugs. We want this to be an entirely curable disease, but I think remdesivir helps move that needle, and it certainly helps people get better sooner. They may be infectious for less period of time. And the sooner they get out of the hospital and are feeling better, the less complications they have. And it's, avoiding days on oxygen, et cetera. So could we do better? Absolutely. And I would love to see us do better and be smarter about this. But I think for right now, for my read of the literature, remdesivir is still a really important part of our arsenal for hospitalized patients on oxygen.
Right. My last question. I think you were part of a panel I had maybe in June or something, and I asked people what would be the game changer, therapeutics or vaccine? And I asked them when that would be. I don't think anybody said it would be by December of this year or November of this year. But I think more people weighed in on therapeutics than vaccines as being the game changer. I forget, I may have to pull the tape and see what you said. Are you surprised by the vaccine news that it seems like it sort of has trumped the therapeutics, because the therapeutics are making a difference, but that it's been so fast and so effective?
I think it's incredible news, and it is amazing when you see all of the same type of vaccine. They're made by two totally different companies, and to find preliminary data that are so similar really, I think, helps raise our confidence in this. Because when we all saw 95%, you think, well, that's great, but can that really be real? And then to see that again in a very similar population, I think really raises our, enthusiasm and belief in that data. We want to see it all, as Paul said. I think your point about therapeutics versus preventative therapies and vaccines is that, we're not going to be able to treat our way out of this disease. It doesn't mean that therapeutics aren't important. We need to save people's lives, and that's where therapeutics come into play. It'd be great if we could treat people outpatient and reduce their infectiousness and how they feel. But in order to really be a full game changer, as you say, we have to prevent this disease from happening. So that's where a highly effective vaccine, I think, will be critical. And then the money is on how do we get this to the people who need it in time. And I think the question you asked me, Bob, was when can we go back to life as normal? And I think I had said June of 2021. And you know what? I think with these vaccine findings, if we can get the vaccines out, including new vaccines to everyone, that may still be true.
That sounds, I just booked a vacation for September, and I may not be alone in doing that. Great. Annie, thank you so much. As usual, really helpful. You take us through this stuff very clearly, and it really is helpful in thinking it through. A lot of very interesting and exciting news as we move toward the new year. So let me let all of you go. Thank you all for joining us. Thank you to our wonderful production team, whose names are listed here. We'll take next week off for Thanksgiving. Please have a safe Thanksgiving and follow the rules. CDC just came out with some new guidance. on Thanksgiving, and it's nice to see CDC coming out with guidance. We'll be back again in 2 weeks, and we'll do another COVID Grand Rounds then. Until then, stay safe.